It looks like we're starting to come together. Thank you very much for joining our webinar today. And my name is Dr. Robert Lyons, and I would like to have a very special guest presented today. Last week we were talking about the microbiomes and cocoon and the immune system and hypoxia a little bit, which later I would like to get in some other time to explain it in more details. But today we have a very, very special guest. Why special? Because I know this professor for many, many years. And when I approached him first time, I asked him about cocoon water and the phenomenon we see. And he just actually told me off. He said, no, it's just only a water. I'm working with the University of Debrecen. Maybe if I have time, check. But again, after a while, I give him some water, and he was kindly enough to start to work with it. He will tell you all about it. And Professor Endre Mathe is not just a professor of my supervisor at my PhD, but he also a very special scientist. And he's working with genetics. And many, many years I've been following him. He has a lot of publications. You can look it up at cocoon.eu/webinar. You can look it up his resume. And without any further delay, I would like to get Professor Endre Mathe on the line. Hello, Professor. How are you?
Hello everyone. I'm fine. It's a great pleasure to be with you, and I'm ready to tell you about some of the stories behind the research that we are doing. I really want to share with you. It's quite exciting and broadens horizons regarding what cocoon is capable on. So tell me if I can start, and I'm ready to give it a chance.
You may start. You may start, Professor. You may start.
Okay, thank you very much. Yeah, it's a great pleasure to meet all of you in the story of cocoon. As Dr. Robert, and also I'm really thankful to Dr. Robert Lyons for involving me into this story. Though at the very beginning I had a lot of doubts about what would be the outcome, but seeing is believing, and for a researcher, if the experiments are telling some results and some conclusions would point into some evidences and some conclusions that would be relevant, then it's pretty obvious that we have to continue and we have to work on cocoon. So today I really want to talk about don't get too scared because of the title. It's about the challenges that life, the interpretation of life would be about based on molecular events. And when it comes about molecular events, certainly we should involve the genes, how the genes would integrate, would control all the life-related phenomena, and how this have impact on a multicellular organismal level. So I wanted to call the story it's the interplay between complexity and simplicity, because I'm being asked always to try to explain things as simple as possible. That's also my aim, but you have to understand that simplicity is also about complexity, and complexity is also about simplicity. And yeah, it's challenging to get to from a very complicated situation to get to a very simplified result or conclusion. It's a hard work, and it's quite challenging. but I'm gonna try today to explain you that though it's really challenging, and the way we are approaching this challenge, it's basically very much specific to where the forefront of molecular biology and molecular genetics is situated. So let's start the story, and I'm trying to feedback all the time to this complexity and simplicity of approach, and to continue the story, moving forward. You see, whenever we combine very simple things, like for instance curves or lines, actually combining in different ways would generate a kind of complexity, a structural complexity. But that's quite simple, because it's a fairly static type of complexity, and it can be seen and visualized, and we can get back to this situation, this structural complexity, because it's stable. However, when we are dealing with life-related events, there too we can say that the atoms, the molecules and macromolecules, and also the organelles, if we put these things together, basically would not result in what would be a cell that would be alive. Why? Because something is missing. Is missing the type of mechanism that would integrate all these atoms, molecules, macromolecules, organelles, that would render these components, cellular components, to work in concert. And what is going to render, what is going to set an order with respect to all these components, is the way the genetic information would get through the system, and is the way the genetic information flows through the system. So it's a fairly dynamic process, and next to this very simplistic structure, combining, you know, some simple structures, we have a cell where we have certain not that many atoms, but through these atoms, through the combination of atoms, we would generate a lot of molecules, and through these molecules, through the combination of these molecules, which is again, which is not a static type of combination or interaction, it's a fairly dynamic. So the dynamism of cellular events would make our life even, and this complexity, I mean, the adjustment of life-related phenomena to the challenges that a cell or a tissue or an organ, or basically a system of organs, or why not the whole organism facing, it's pretty challenging. But somehow the genetic information, the way the genetic information flows, or why not to say the way the genetic information is expressed, or the genes are expressed, or the genome is expressed in different type of cells, would set an order, a proper order. So life is very much about, so the new dimension of the life is very much about setting an order inside the cell, and this order or this hierarchy or structure, dynamic structure, is absolutely relevant for the well-being of the cell. Good. So we can also say, in a very simplistic way, if we were to simplify what life is about, life is nothing else, and you have to excuse me, because I'm a geneticist, a molecular geneticist, and a developmental geneticist. Okay, so everything related to genetics. So you can say that this is the genetics would be my glasses, and I would see you know, the world through this type of glasses. But it's actually genetics is the life sciences that brought us the cause-effect type of correlation in the reach of our hands, when it comes about explaining life-related effects. So life, getting back to the genetic information, the flow of the genetic information is about expressing or setting the gene expression or adjusting the gene expression with respect to the environmental challenges. This was quite a surprise for many of us, many geneticists, because we were very much considering, the genetic information a kind of material that is fairly stable, and the stability of genetic information is absolutely crucial for inheriting the genes down the generations. That's why, many of us being interested in the inheritance, in what would be the classical meaning of inheritance, we thought genes and the genetic information is very stable thing, and this stability is passed down the generation, and based on this stability, many other things would take place, and life is going to be nice and wonderful. But it turned out in the past 20, 25 years that life is very much controlled through the action of genes. The way basically these genes are going to intervene, and the intervention of genes is mediated through the expression of them. So you should imagine, this kind of gene expression like reciting a poem, and based on the way, you recite the combining different tones, rhythms, intonation, pronunciation, the outcome of the poem could be fairly different. And that's how we should imagine, the expression, the outcome of gene expression modulation. And that's really, that's a really amazing feature of life when it comes about explaining on a cellular level, when it comes explaining life on a molecular level. You will see today some other challenges. I'm going to bring it forward regarding, how gene expression generates all this complexity. So actually, we are facing a situation when we redefined, when we are redefining, we are giving a new definition to life, and this definition is based on a system biology type of research strategy, which basically sees or envision life or any kind of trait related to life, or any kind of mechanism related to life as the sum of the interaction between genomes and environment. So then when it comes about environment, you can imagine issues like nutrition, challenges like physical, social, mental types. And all these things certainly would have an effect, would generate an effect on the genome. And this effect is gonna get materialized in specific gene expression profiles. So actually behind every trait, and you name it, whether it's health, whether it's disease, whether it's aging, whether it's hypoxia, whether it's obesity, or diabetes or cancer. So any trait that you can imagine should be envisioned as a phenomenon that develops in time and that is adjusted in time. And this is based on how the genes that participate in getting materialized the phenotype, how the gene expression is taking place. So the idea that in order to understand the cause-effect type of correlations behind every trait, behind every disease, behind every, let's say, feature of humans. So the idea to look for the genes and the way the genes are interacting and in the protein encoded by the genes, how they interact would let us understand cause-effect type of correlations. This is a kind of new approach It's very much forced by us geneticists. Why? Because genes do not lie. And whenever genes get expressed, they would tell something to the system, tell to the whole body what to do, how to react upon some environmental challenges. Yeah, it's working, and I can tell you that it's working. During the past 20, 25 years, why not to say 30 years, researchers moved on and basically not being absolutely aware of the importance of the gene expression, nevertheless being interested in how the genome works. There have been developed new methods, and we are saying that right after, in this new century, we step in the so-called omic era of life sciences, where basically we developed really powerful methods in order to follow the flow of genetic information in life-related systems. And among these methods, some of the methods would address issues related to the genomes, to the epigenomes, why not to say the metabiome. Last time, the previous webinar was about metabiome. We really want to analyze the metabiome. We can use epigenome type or, genome type molecular methods. Then it comes to transcriptome, how the genes, when the genes are expressed, basically their expression is very, and taken together when we are referring to a cell, or why not to a whole organism. The totality of the expressed genes would materialize in what we are calling the RNAs, messenger RNAs, tRNAs, ribosomal RNAs, microRNAs, long chain RNAs, and so on. So we can analyze all these, or the whole transcriptome of a cell, the whole transcriptome specific to a whole multicellular organism, so at individual level. Then the next stage, right through the combination of RNAs, and also amino acid proteins are going to be produced. So we start from the genes, then we go to the transcripts, and then from the transcripts we get to the proteins, and the whole set of proteins that get formed inside the cell would we call the proteome. We have some methods by which we started to analyze the proteome, but our methods currently are not that proficient. They are not that efficient in englobating data regarding the whole set of proteins. Metabolomes would be another set of molecules that we find inside the cell. These are the metabolites, the intermediates of some metabolic pathways. We have methods to analyze. So you see, we can generate a lot of data regarding how the genes in the genome are expressed. And so why not to do that? Why not to do that? We really want to know how genes are controlling events inside the cell, or how genes control events get modified upon the action of environmental factors. We can address such questions by analyzing the whole set of genes, the whole set of transcripts, and eventually we will reach the level to analyze the whole set of proteins. Is this really important to analyze, or would it be really important to analyze all the genes that get expressed, in a cell? The cells tend to achieve a kind of steady state, or equilibrium, or harmony, or homeostasis, you name it. And even we can move it further up to the level of the whole organism, so the individual level, which is composed of many eukaryotic cells. And yes, and certainly we have learned in the school during our studies how this homeostasis is achieved on the level of the whole body. And we, in this homeostasis or steady state or equilibrium, were implicated, the hormones and neuronal type of regulations. So in a very simplistic way, basically we can say that, and choosing the sample as a topic, the blood glucose level, it's very simple to envision that we have two hormones, the insulin and the glucagon, and the interplay of these two would regulate, would set the normal blood sugar level or glucose level. But this is a very simplistic way to talk about a phenomenon like the blood glucose level regulation. However, if we really want to get into details, then we will realize that there is a specific organ, the pancreas, and inside the pancreas there are specific cells in some compartments of the pancreas, where basically inside this compartment two types of cells would be challenged and would be capable to synthesize either the insulin or the glucagon based upon the concentration of glucose inside the blood. And when it comes about these two hormones are again synthesized by genes, and these genes are placed in these two types of cells in the Langerhans islets, and they can sense the difference and take action. So basically, you see, to explain the blood sugar level regulation, we have to implicate some specific cells, and inside these cells, specific genes. So we started with the physiology of blood sugar regulation, and basically we ended up to explain what are the causes of what we see on a physiological level by implicating genes, implicating specific cells and specific genes. So yeah, that's complexity. So it's a simplistic way, but now we are moving to the molecular explanation that would complicate things. But and also I have to admit that at this stage, basically molecular data are really complicating our life, and basically we are looking for solution to get the message, the main message, the take-home message out of this molecular data, because we are hoping that finding and defining new methods in order to interpret the molecular data would bring us much closer to explain the cause-effect type of correlations behind cellular events. And actually, the biology type of strategy, research strategy, is depicted on this slide, and you see it's the so-called the magic cube. The magic cube, which shows on one side the experimental systems, so cell cultures, small animal models, non-human primates, clinical studies, so everything, all the possible experimental systems are combined in order to gain information with respect to a phenomenon. But then you see we are applying experimental systems, but these experimental systems are challenged through high output data, genomics that I was mentioning earlier, transcriptomics, proteomics, lipidomics, metabolomics, metabiomics, all these kind of high output data methods would generate hundreds, thousands of data, so much data that we hardly can handle. And for this, we would need bioinformatics, type of data mining or data analysis, and combining these three elements, the experimental systems, high output data generating methods, bioinformatics, we are reconsidering now what would be the proper definition of health, what would be the proper definition of aging, opacity, cancer, type 2 diabetes, metabolic syndrome, and neurodegenerative disease, just to mention some of these, some of the challenges that we are facing. So we started by saying that genes are so relevant and the expression of the genes are really important. And now, I'm telling you that through this very simple approach, we can generate a humongous complexity. And then from this complexity, now we really want to we want to get again to a simplicity, to simplify things. And what simplification would be about, and how we can simplify things. Basically, it's again by putting into this frame the flow of the genetic information. You see in here a very simplistic way of bridging, the Earth with the sky. It's the vision of a sculpture of the Brunkush, a very simplistic sculpture, who basically tried to simplify and try to reach the quintessence of the things. And basically, we would need such approaches, in interpreting, in discussing the molecular meaning of the cells. Actually, what we really need, we would need to think different when it comes about the complexity of life. And that's a huge challenge, because you know very well, bringing new ideas, new data, putting forward in science is not an easy thing. If I were to ask, people from the street whether the traits are inherited or not, 99% people would say that traits are inherited. Actually, we know, and basically the father of genetics, Mendel, in 1866 demonstrated that basically the traits are not inherited. What is inherited, that's basically the gene, not the trait. Nevertheless, this information, this law of, or this truth of life didn't reach the level of people. So it's not that easy to bring new ideas and to propagate new ideas. But this is through new ideas we would manage to make progress. So we have to think about life and about the complexity of life in a different way. And what this different way or innovative way would be about, it's to concentrate on the expression profiles of genomes. So we want to understand what is going on in the cell. Let's analyze, the expression, the whole expression profile of the cells, and not just the cells, but in the case of tissues, organs, and whole organism. And then afterwards, based on the relevant genes, the relevant expressed or significantly expressed genes, to design, to put together a kind of protein interacting network or interactons, and based on these protein interacting networks, to figure out what the phenomenon would be behind this interacting protein network that is gonna get put together upon the expression profile of genes. That's a huge challenge again, and would require and would change all the way we are doing research and the way we are interpreting, life-related phenomena. You should imagine that, or you should accept the idea that the future medicine is about revealing this complexity behind any disease, and then when a diagnosis is put, basically the diagnosis should be based on how the genes are going to express. And by doing so, you can also get into the dimension of getting the diagnosis and also the treatment personalized. So these are huge challenges. But can we really measure and can we really study the expression profile of genomes in the case of human cells, in the case of a living human organism? Can we just retrieve the tissue or the organ from the human body and from the living human body and then check it how the genes are going to get expressed? No, we cannot do that because you know very well ethical and moral and other issues should be considered. And because we are facing this dilemma, we looked for alternative solutions. And the alternative solution to address, to study the way the genes are working together in order to lead to a situation which would be a healthy status, or would be, you know, a diabetes, we would need to focus our attention on what would be the model organisms. Model organisms were always close to us, and we really worked on these model organisms, even those times when we didn't know what would be the link between, for instance, a yeast cell or a yeast species and the humans. Nevertheless, these model organisms, we acquired a great deal of knowledge studying these model organisms. But when we reached, you know, the genetic level, I mean, when we started to look for the genes that get expressed, you know, in the case of some conditions, mutant conditions, for instance, then we realized afterwards, and we described, you know, these genes functions upon using, you know, the model organisms like yeast, like zebrafish, like mouse and frog, and identify the genes behind these effects. Then we looked whether these genes can be found in humans, and it turned out that, yeah, many of the genes that exist in humans and these model organisms are pretty similar. So we share a common pool of genes with these kind of model organisms. So studying how these genes are working in model organisms is much easier than on humans. But nevertheless, if the genes are the same, basically, and we find out what the function of these genes would be in the case of Drosophila, roundworm, or mice, or frogs, that would bring us much closer, that would create us clues regarding where to look when a gene is checked in the context of humans. And this was, again, a very fortunate situation to say, because we didn't know initially that we share a common pool of genes. We were just working on these creatures because we were able to develop methods to do experiments. But then, with the advent of molecular genetics, we realized that really there are common things, common pool of genes. So why not to get even harder into these studies? What this slide is telling you about is basically the similarities between humans and the most loved, adored model organisms. Just Drosophila, if you look at Drosophila, you see basically 50% of the genes that exist in Drosophila have their counterparts or orthologs in humans. And it's also very interesting that 75 human diseases can be studied, and the genetic control and the genetic basis of human diseases can address Drosophila. How interesting, how interesting. And there's also the data are there for other model organisms. I'm emphasizing the relevance of Drosophila because I am studying, you know, diabetes, I'm studying inflammation, I'm studying Cocoon's effect, I'm studying the effect of plant extracts, you know, on Drosophila, using Drosophila as a model organism. Why is Drosophila so suitable for such genetic studies? It has a very simple life cycle, and also it was quite easy. We could induce mutations in Drosophila, And those mutations would lead to certain mutant phenotypes, and based on those mutant phenotypes, basically we could isolate the genes that got mutated, and then once the gene that produced, once mutated, produced a phenotype, then the gene could be used, to check if the gene has its own counterpart in humans, in mice. So basically, we were doing a kind of translational research approach. We were studying Drosophila and then switching to humans based on the genes, the same set of genes. Drosophila offered a great deal of knowledge to what would be the genetic and the molecular basis of life. Here are some people listed for their achievements who were awarded the Nobel Prizes just in 2017, basically 3 years ago. The genetic control of circadian rhythm, 3 scientists got the Nobel Prize. They were using Drosophilas in order to demonstrate, to identify the genes behind regulating the circadian rhythm. Nowadays, we know that even the drugs that we take in the case of some treatments should be considered with respect to the circadian rhythm. But then, when it comes about the innate immunity and the genetic control of innate immunity, in 2011, 9 years ago, Julius Hoffman and Bruce Butler were awarded the Nobel Prize because they to describe what are the genes behind innate immunity. So you see, it's and also there are about 30, 40 research teams across the world who basically came together and organized a consortium working on Drosophila, on C. elegans, zebrafish, in order to study rare genetic diseases. If the rare genetic diseases specific genes do exist in this type of model organisms, they are very much propagating what we call the translational type of research. So we can see that the grounds, the scientific grounds for the translational research would be the common set of gene pool. But you can say, okay, we have the same set of genes, but do our organisms and the anatomy of our organisms look similar? And here it is the answer. Yeah, there is resemblance regarding the structure or the anatomy of the body. You see the colors would represent the corresponding elements between flies and humans. And also, if the structure is pretty similar, the genes are similar, we can also envision that the mechanisms, the molecular mechanisms regulating, or integrating the information at the level of the whole body would look pretty the same. And indeed, nowadays, this kind of translational research is getting very much promoted. Why? Because we can gain, using model organisms, we can gain a lot of information, a lot of relevant information regarding how diseases, how certain conditions get developed, appeared, and what other features of these conditions evolve during the lifetime of an individual. And from flies and other model organisms, we can switch to humans, and we can ask direct questions. I mean, if the gene is implicated in the autism, good. Okay, does that gene have a counterpart in humans? And if it has, then what is really happening with it, and how is gonna behave that gene? How is going to get expressed with respect to autism? So it's not like a blind type of approach that we have this step, in a room where there is a total darkness. We have signals that we can follow and ask proper questions. So I was trying to explain you what are the challenges that we are facing in the case of putting life on the grounds of genes functioning. Now we would move on and switch to what would be the cocoon story. And Dr. Lyons was telling you earlier in the introductory part when he introduced me that I was not that sure on me when he approached me asking, would you be able or would you like to do some experiments on cocoon to see if it's going to affect anyhow Drosophila? And I said, oh, okay, come on, this is a water, so I'm not interested. A water is a water. What difference can make a water? But then it turned out basically that I did a very simple experiment, yeah, using fruit flies. So I was feeding them normally, and in the media of the fruit flies, certainly we have to we have to add water. So I set two types of media, so one with normal water and one with cocoon water. On the graph, you can see the red line corresponds to the normal water, the blue line corresponds to the cocoon water, and the graph shows the lifespan, you know, of the population. So actually, you can see something pretty obvious. For instance, on day 25, the red line peaks suggest a situation when basically 60% of the individuals were alive. If we look at the cocoon point, that would indicate 90% of the individuals being alive. So that's clearly indication or a suggestion that basically upon cocoon, so when the flies were drinking in the media, were having cocoon water, the average lifespan increased. I was quite shocked seeing such an effect. So I said, okay, let's just repeat the experiment. I repeated myself a couple of times. Then I asked my PhD students and undergraduate and graduate students. A lot of people have repeated the experiment, and we could see this difference. The cocoon was increasing the average lifespan. This was the result that made me very curious about cocoon. Then I said, okay, so why not to complicate things a little bit, and let's see what we can what cocoon can do in more complicated experimental setups. And these experimental setups were about, you know, challenging the flies on a normal media or a high sugar media, or applying hypoxic shocks or normoxia type of combination. You can see in here, you know, just not giving too much details, just the relevant part. Whenever, you know, the flies were kept, and not just the flies, the whole life cycle of the flies went on a media that was containing normal sugar, so normal media, normal water, and we exposed them to a hypoxic shock. 22% and only 22% of the larvae survived this hypoxic shock and reached the adulthood. When we redone this experiment with the media that in which the normal water was replaced with the cocoon water, 84% and applied the hypoxic shock, 84% of the larvae survived to adulthood. That the cocoon water somehow can rescue, can decrease the hypoxic shock viability reducing effect. It's clear, 22 from 22 to 84. That's a humongous increase. When we repeated, we did the same experiment, but this time, you know, we replaced the normal media with high sugar media and the hypoxic shock. Only 20% of the individuals survived. Replacing the normal water with cocoon water on a high sugar media, we got a survival rate increased to 72%. So clearly, the cocoon rescues hypoxia, the detrimental effects of hypoxia. We were really happy with these kind of experiments, and then we said, good, it's nice. Let's just complicate ourselves the experiments and focus very much on what is really going on in the case of high sugar medias and combining with hypoxia, high sugar just alone with normoxia, and high sugar in combination with hypoxia, and then introducing the cocoon in all these experiments. What we got from these experiments was something really interesting, namely, so when basically we were subjecting the individuals to this high sugar media with normal water, we could see that there will be a developmental delay. So the larvae would stay 10 days in this stage because they are struggling to cope with the high sugar effect. I can tell you that the high sugar effect generates inflammation, a very strong inflammation, and and many of them, basically very few of them, just 10% would survive, would reach the adulthood. However, when we were replacing the normal water with cocoon water on a high sugar, you do remember survival rate went up to 72%. And also, quite interestingly, the developmental time for the larval period, shown in yellow, got reduced to 8 days, from 10 to 8 days, suggesting that the cocoon, when we are applying cocoon, the cocoon can overcome the high sugar-induced developmental delay, which basically is a very strong inflammatory state or status. So the question is, very complicated experiments, but to get the meaning of them, we can ask some very simple questions. What is the reason for the cocoon-induced partial rescue, or in the case of hypoxia type of experiments? Or what is the reason? How can what cocoon is doing to get reduced the high sugar-induced developmental delay, or why not say inflammation? Again, to answer these questions, we can go back to this system biology type of paradigm of life. So again, how to combine the genome with environmental cues, and then the specific gene expression profile based upon the specific gene expression profile to explain, this hypoxia reducing effect, which could be considered a trait, or this anti-inflammatory effect, again, could be considered a trait. What we have done, we applied the transcriptome analysis. So we analyzed all the genes, how the expression of the genes has changed upon these kind of treatments. And we got some very interesting results. Again, a very complicated scheme or graphic, but we are not gonna get into details. Just the main relevance, okay, would be, that in these experiments that I was telling you earlier, I was presenting you earlier, we could see that about several gene expression profile has changed. The arrows are indicating the genes whose expression got increased 2 times, 3 times, 4 times, but because this is on a log2 scale, you can imagine that log2 would correspond to a 4-time increase, and so there are huge changes. On one end, we would have an increase, and on the other end, so the low value domain would correspond to genes whose expression really decreased several times. What these experiments are telling us would be, that there are not just a few, but several genes, very many genes whose expression is going to change significantly when it comes to explain the previously formulated questions. I mean, how the cocoon rescues hypoxia, how the cocoon reduces inflammation. Several genes are implicated, and it was a quite of challenge for us, because we had to take every single gene and verify whether the change and check them out. And we got, from the experiments, from the sequencing experiments, RNA sequencing experiments, we've got, you know, a raw data, such tables as you can see in here, and then the colors we put ourselves, but the colors do represent what the gene would do, what the function of the gene would be about. Okay, and you can see, for instance, CG 13040, that color corresponds to the cuticle type of functions. Cuticle type of functions are related to the carbohydrate metabolism. There are quite a few. There are quite a few. Other colors would stand for signal transduction. Other colors would stand for immune response. The pinkish colors would indicate genes involved in the immune responses. Okay, so but then you see there are about 15,000 genes evaluated in every single experiment. Okay, and certainly to go all the way through these 15,000 genes, it's fairly time consuming. We did that, and actually we were able to set these genes in roughly in 20 clusters. Here they are shown just 15 regarding the function of these genes. So we have genes whose expression got changed dramatically, and the function of these genes would be related to the amino acid metabolism. There are other genes involved in lipid metabolism, carbohydrate metabolism, mitochondria-related functions, cytoskeleton, extracellular matrix, oxidoreductases, stress, immune response, signal transduction, transcription factor, cell cycle transduction. So a lot of that was quite a surprise for us. I mean, we knew from these graphs that there are several genes, several hundreds or thousand genes implicated in this cocoon-generated hypoxia rescue, in this cocoon-generated anti-inflammatory effect. But how to set an order in these genes, how they are related to each other, because you cannot imagine a situation where every single gene would act independently from all the other proteins and all the other molecules existing in a cell. All of these proteins are interacting and would find partners, and through these partners would generate, you know, their effects. So the first to set a kind of to analyze the relationship between these genes and these gene-generated proteins, we applied a principal component analysis type of statistical analysis or evaluation. And here you can see different colors. The different colors again would represent genes that have specific functions, like I was telling you earlier, related to some metabolisms or related to the cytoskeleton and so on. And you can see basically that most of these genes are clustering together, would form a close cluster. However, there are some genes that would stick out. For instance, you can see the LSP1 gamma. That's a gene that encodes for larval serum protein 1. Next to it, there is the larval serum protein 2, and then there is larval serum protein 1 beta. So what these genes are about, and then there is the larval serum protein alpha. These genes are about would synthesize proteins that are absolutely relevant for the metamorphosis of Drosophila. That's going to happen during the pupal stage of development. And we know that these genes are expressed to a very high extent, suggesting that upon cocoon, so one of the cocoon-generated effects would be to increase the protein stores, the stored protein amount in the larvae in order to facilitate metamorphosis. So what would be to take further this conclusion? We can consider this data as an evidence for forcing. I mean, cocoon is forcing, is increasing the extent of protein synthesis. Okay, so you can imagine that there is a humongous set of data that we are still analyzing. And the past 1.5 years, basically on a daily basis, I spend 2-3 hours on analyzing this data, and I'm absolutely fascinated by the outcome. And I have to tell you, I have to put forward the explanations or the answers for the questions. So Cocoon is inducing gene expression changes that can be put in, that we can put in the context of metabolic profile adjustment or mitochondrial ATP production or protein synthesis and folding and also protein degradation, how transcription is regulated, then the cytoskeleton, then extracellular matrix, and anti-inflammatory effect and innate immune response. It's quite interesting, and you could say that while you were studying genetics, where you were taught genetics in the schools and during your studies, all the examples were about one gene, one trait. Now I'm telling you that there are several hundreds of genes implicated in several traits, and these traits coming together would generate even complex type of traits. And we should imagine, you know, basically diabetes and cancer and all these things like the combination of very complicated traits. I just wanted to give you two brief examples of what Cocoon is doing, because we know what we have identified the genes whose action is going to get modulated upon a Cocoon. For instance, we have here a situation that is very specific to any kind of hypoxia. Whenever a hypoxic situation appears in the cell, the first thing to happen, you know, the first really dramatic effect to happen would be the reduction of ATP utilization. Why? Because there will be no ATP produced. Why there's not going to be any ATP produced? The reason for that would be the fact that a mitochondria is going to produce a lot of reactive oxygen species, and these reactive oxygen species would increase the synthesis of AMP, adenosine monophosphate kinase, and this enzyme is going to get increased in its expression. Basically, is going to inhibit the protein synthesis through translation, and also the synthesis of sodium potassium ATPases that are absolutely relevant for the regulation of the membrane potential. However, when Cocoon is put in action, the Cocoon is going to inhibit the ACC. ACC stands for an enzyme implicated in the synthesis of fatty acids, among others. And this gene, once it's going to get expressed at a higher level, through its action is going to inhibit the AMPK, and AMPK getting inhibited, its inhibitory effect is going to get abolished, and basically translation and the sodium potassium ATPases activity is going to be maintained. So basically the cell, it's a kind of mechanism by which the cell will manage to survive the negative effects generated upon hypoxia. You can see here other more complicated image related to the Krebs cycle and how Krebs cycle is modulated. You can see that the acetyl coenzyme A is going to get very much pushed towards what would be the fatty acid synthesis, and also you can see that the FA and the fatty acids, once the fatty acids are produced, they are gonna get pushed into beta oxidation, so they are gonna be used for ATP production. So here we have some genetic evidences, okay, upon which we can explain what I was telling you earlier, namely that translation is not going to be inhibited, and there will be enough ATP produced to support these activities, even in the absence of oxygen. Now, because we are living in interesting times, and the COVID issue is a huge challenge for the whole humanity, the cocoon, we were able to identify a lot of genes that are implicated in the immune response. So in Drosophila, we have a set of genes called AMPs, antimicrobial proteins. These genes are really related to what we would call the innate immunity. And you see there are of different types like drosomycins, diphtherisins, IM1s, and these expression, their expression is gonna get changed, but certain genes like the IM33, which is absolutely relevant for fighting against bacteria, its expression is going to increase really dramatically. Then other genes like the DUOX and the IRC, the iocase complex, again involved in the oxidant detoxification and regulation of the gut microbiome, are expressed at really dramatic levels. Then we move to the lysosomes type of genes, B, C, D, E, S. They are really efficient to find the gram-negative bacteria. Their expression again are going to increase heavily. And then we are the RM62 and the RANSEC genes, which are used in the antiviral and antimicrobial response. These two again are going to get expressed at a high level. Then we have some shock proteins, some of them like 23, 26, 27, 67, 68, 83, and these are also indicated in the immune response. Again, their expression is going to get significantly changed upon cocoon. Roughly, what we can say, or what I can say, and I'm pretty confident about it, cocoon is modulating the innate immune response, at least in Drosophila. Whether it's going to have an effect on the COVID, for instance, I cannot tell you. We would need to do to infest, Drosophila with this virus. But again, there is the problem whether the virus can have an effect on Drosophila or not, because it's the receptor story. If it has the ACE2 type of receptors, I don't know that. I have to check. I'm not aware of the details regarding the ACE2 receptors. If Drosophila has on their endothelial cells such receptors, then indeed the COVID-19 would be able to induce some infestation. Nevertheless, what I can tell you, and I'm pretty confident about it, the immune response, the innate immune response, is going to get modulated upon cocoon. So we would need to do some experiments on human cells, using cultured human cells, to address this issue of the COVID-19 infestation. When it comes about the hypoxia, reducing hypoxia and the hypoxia generated the negative effects, yeah, I can tell you Drosophila is responsive to cocoon, and cocoon can reduce these kind of the hypoxia generated effects. And to see and to bring to take you further into these studies, here you can see some interactions. The way the interactions for the clusters that I was telling you earlier, and I mentioned that there are 20 clusters regarding gene functions, how these genes would interact with each other upon phenomenon. And this interaction is depicting, genes implicated in the muscle structure, genes implicated in heat protein, genes implicated in glycolysis, so metabolism, carbohydrate metabolism, genes implicated in fatty acid metabolism, and you see the links. What are the links? What every single dot stands for a protein, for a gene encoded protein. So what would be the next stage for us? Basically to put together such interactons. I mean, how the interactons, how certain genes encoded proteins would react with each other. Why we can do this? We can do this because actually for Drosophila, for every single Drosophila encoded protein, it has been changed, whether it has been checked, analyzed whether that protein is going to interact with all the other protein types that exist in the proteome of Drosophila. So that's terrific. That's terrific. So we would be able. What is this interactome telling us about? It's basically is the protein-protein interaction network in the muscle fiber development and function. So basically, you see through these interacting genes or through the proteins that are encoded by genes, basically we can define how the muscle develops, and getting, you know, looking at this network, basically you can imagine situations, you know, when one of the elements, one of the genes are going to get expressed to a higher extent, then that that gene or that protein is going to inflict an effect on that network. And if that network is enough stretchable, then, you know, the outcome could be that basically the muscle fibers are going to get developed at a faster speed or to a higher extent. Or if the network is stretched up on a particular gene and it's going to get defective, and the whole system could collapse. So that's the future of what is ahead of us. And basically, as I was telling you, we have identified the genes. We know, and now we are and we will assemble such networks, such interactons, and we would define the most sensible or the most relevant genes inside these networks, and that would give us the certainty about the proper effect of, and how, you know, the effects would or different kind of phenomena would cooperate in generating the final outcome. So Cocoon is able, because I was emphasizing at the beginning the translational type of research and the relevance of the translational research, you see we are on this pathway. We started analyzing Drosophilas. We know we were able to observe multiple effects regarding gene expression, and also we were able to identify or to show that these kind of effects, that the Cocoon-generated effects, are very much dependent on the nutrient and oxygen availability. Now that we have the players, the genes identified, and we put, you know, into these interactons, we would know what to look for. And basically, when it comes about humans, we would just in a very targeted experiments look or check if those genes indeed are relevant for the Cocoon-generated effects in the case of humans. But when it comes about humans, we will face another impediment, a very relevant impediment, namely that there are no two identical genome containing individuals, with the exception of monozygotic twins. In Drosophila, it's fairly easy because we can generate individuals that have the same, several hundreds of individuals that would have the same set of genes and the same alleles for every single gene in question. So basically, they would be twins, and then the problem of genetic diversity is not going to interfere with the outcome of our experiments. So getting personalized and getting the cocoon effect personalized in the case of humans, that's going to be another huge challenge for us. So to close down the story, our efforts are directed to explain the cocoon-generated effects as simple as possible, but not oversimplifying the complexity of life, because getting oversimplified, we would lose the meaning and the relevance of the cocoon. Thank you for your attention, and I hope you could follow me. I'm just too excited about what cocoon is about. I'm pretty sure that in the following 10 years, I will have things to do. I will have plenty of questions to answer. Thank you for your attention, and I'm ready to take questions. Thank you.
Okay. Hello. So thank you very much, Professor. It was great. It was really, you went through much easier in the course like you went with me. And I think we need to see this several times to understand, but
yes,
underlying really is cocoon water has effect on drosophilas and has a gene expression. Nobody can say now no. So we have,
yes.
this is what I get from the presentation. Luckily, I heard this several times, and I believe in the future is that we will need to publish some papers about this, and we need to go and find more results and find more fantastic, for you say, exciting elements of this study. And now we are open for questions. Yes, yes, Professor, yes.
I just wanted to emphasize that everybody is talking about evidence-based books. I mean, cocoon is inducing changes. That's the ultimate proof. That's the ultimate evidence, that has some effect on life. So measure whatever you want, but the ultimate proof for having an effect is the changes that are induced in the expression. So we can be absolutely sure about this, that is interfering with life, cocoon. Okay, and as I was showing, it could be quite, these kind of effects are quite favorable, are in the sense that in some unpleasant situations or pathological situations like hypoxia or inflammation can get the system back to normal. It's pushing this interacting network of proteins regarding inflammation, regarding hypoxia, is stretching to what would be the steady state, what would be the normal state. I'm confident about this.
Oh, that's fantastic. Thank you so much. Thank you very much for everybody. And thank you very much, Professor, to be with us this evening. And now we are open for questions. If anybody have question, please do. We see the wall here. We had one question not relating to the professor, most likely on the breathing techniques. It's been worked for thousands of years, especially in Eastern medicine. We call it Qigong or breathing exercises. And we know they really can make their life longer if they can hold their breath and then breathe in and out and do meditation with it. But also, it's close to the ideal of cocoon. That was, I think, the question is, or the statement on that. And I just want to state it. Cocoon water does elevate oxygen pulse or pressure in the tissue, not mainly the key to elevate saturation. Breathing, it's bringing oxygen and binds it to hemoglobin in the lungs. But cocoon water bring oxygen to the cell immediately without delay or without distribution problem. Even if the cells wants it or not, the oxygen will get there. It's due to pulse or pressure different. So the idea is the same, just like hyperbaric chamber, but it's too high, cannot be too long. We done studies with breathing exercises to try to measure oxygen pulse or pressure elevate. We could elevate saturation, but we could not elevate oxygen pulse or pressure. Just give you an example. If you have a wound in your hand or your leg or anywhere in your skin, you cannot just sit there and do the breathing and the wound heals up. That's not possible. So you need to get much more oxygen to the cell to start to repair and bring oxygen to the tissues. Otherwise,
can I just add a comment, because
yes.
you were saying that bringing oxygen to wound healing is really a great thing, and I can tell you that based on what we observed regarding gene expression in the cells that are at the edges of the wound, okay, at the margins of the wound, the cocoon would increase the expression of cytoskeletal genes, the actin genes, the myosin genes, some tubulin genes that would mediate, that would facilitate wound healing. We know the molecular aspects of the molecular control of wound healing, and it's upon cytoskeletal changes, and cocoon facilitates that. So we have the proofs on a molecular level demonstrated upon gene expression. So we will be able to come up with many demonstrations how the wound healing is facilitated by cocoon on a molecular level. Sorry for intervening.
No. This is very good.
But we will have such many such proofs supporting what you have seen, what you have observed on the physiological level.
Yes, we have thousands of those, so we see that every day, and this is very good what you're saying, because really supporting what we see.
Exactly.
So that's what we see. I want to show you some of the let's say I will show you one on this one, on the yes, the lábszárazat jöttök. Yes, ezt akartam pont. Okay, here is a case. You can see it soon. That's what the professor say, and that's what I'm seeing in the other end. And this patient had a ulcus in his feet, and then you can see the necrosis on the leg. The veins are really necrosized, and the surgeon just cut it, and eventually get deeper and deeper, and they have nothing to do with this. So if they would just follow and cut more, and be somehow not helping the condition of the veins, there would be no way to help. So in order to bring oxygen here, you need to bring it directly to the wound, not just breathing or go to the chamber. You must be in there. So you can see the healing begins, the margin, right? There is the margin. You can see around the wound. It's like a pink margin. And you can see the vein conditions is very mild now. So the inflammation effect of the water take place. And then we can see what's happening with it when completely disappears. So that's a very good example. You need to have a right amount of oxygen through the skin, even to the wound. An open wound, it heals up very quickly. And that is the complete oxygenation of the cells. So it's not just the surrounding, everywhere there is oxygen. And the breathing exercise is very good, and I would really recommend to everybody, because I've done it for myself and I do it myself too. So that would be my answer. So other question here is, thanks for the great info. I would like to know the beneficial effect on the Droser studies. Professor, can you see these questions? Can you see this? Can he see
Yes,
it?
yes, yes.
Okay, so
yes.
please, maybe you can answer.
Just a second. I'm reading now, if you excuse me.
How much cocoon water? What was the amount of the cocoon water given to those studies? That was also, it's a very good question. How much water they get when the study went on? Can you see that?
Actually, I am trying to get but can't really get it. But I can tell you, basically, in the case of Drosophila, we have, not we, but other Drosophilists were able to work out to define all the nutrient needs of Drosophila regarding normal development life cycle. So we have the values defined for every single component of the meals that we have to provide Drosophilas, and also the water. The Drosophilas develop properly in when humidity is about 70%. And basically in the food we put not more than a total, let's say not more than 10%, 50%, 15% of water. Yeah, so it's not like and the media is fairly solid. It's a kind of jelly stuff that would contain about 15% water. It's not a huge amount to but that's enough, and that's okay.
Mm-hmm. And how often are you changing the water? I mean,
I'm not changing at all, because the life cycle of Drosophila is about 10, 12 days, and from these 10, 12 days, about 4 days they are not going to eat anything. So it's basically 6, 7 days when they are eating, and they are using water. So basically the water is there, we put in the media, it stays there while the Drosophila develops, goes through the larval stages, and when reaches the pupae, from that moment on, it's not going to use any water. Yeah.
This is very interesting, because that's mean a very small amount of cocoon water keeps the oxygen very long time.
Yes.
It's already proven how much oxygen can stay, because usually oxygenated waters, if you leave them open for an hour or 20 minutes, 30 minutes, all the oxygen is out of it. It's not staying inside there. But it seems
to be the case of
12 days. What?
But it's not true for cocoon that the oxygen is going to leave in few days, the water, so it stays.
Yes, we measuring this water constantly and averaging about 3 days. If somebody leaves it open on the bottle for 3 days, the oxygen partial pressure is the same, and the saturation milligram per liter is always the same. So it's very easy to. We do a lot of measurement on this, so that's why the question was very valid. So we use small amount of water but highly oxygenated, and the oxygen stays stable. Currently,
Exactly.
you're saying right now for 10 days, 12 days.
Yes.
Okay,
Yes.
anything else? Okay, so tonight I have, from my heart, thank you very much coming to join with us, and thank you all of you who come to listen all the way. I know this is going to get more smoother, and we can try to work on the quality of the internet also. In Hungary, it's a big storms now, and it's changing all over, but we are trying to bring in better quality sound, and we would like to invite you back to maybe specialize in small things. So nice that overall we understand, and specializing on detailed let's say, hypoxia and cocoon relation, and or immunology effect of cocoon relation. Right now, we're working on actually we were publishing right now in United States Health Magazine. I will send it in the internet if somebody interested to read about it. We will have a article on the way how the Hungarian Ayurveda, homeopaths, naturopaths, how they think about fighting COVID-19. And they put together a protocol from Semmelweis University, and then they put it out, and now it will be published by one of the professors from there. One of them, namely, I know, Ivan Szalkai, PhD, MD, who does all these Ayurvedic techniques also. And here's another one. Have CO, yes, there's a question here. We have, it says, have COVID-19 patients been given cocoon water? I tell you, for the last 2 months, I've been sending water to several hospitals, to the doctors and their patients. We give them the water free, so they can fight in the line of fire there. And mostly we have very good reports. And cocoon water is not specifically treatment for COVID-19. Cocoon water mostly helping the immune system functions better, namely the B cells, the T cells, the natural killer cells, and their functions. That's what is supporting. And as we heard today from Professor Endre Mathe, the hypoxia issue. And now is more focused today, more focus on the hypoxia issue than the actual viral infection, because it looks like the viral infection is just the beginning, and there's a bacterial inflammation in there also, which is causing the big problem. That's why when the lungs start to collapse, and now from Italy, there is new studies coming out, just came out today, they said and stated there is other effect on this. So we have to look at, the effect on the thrombocytes. Maybe there is something correlation in the viral infections and thrombus developing in the blood and getting to the lung and causing hypoxia. So there is a lot of interesting new things. This is a new virus, so we don't know too much about it. So everybody trying to get as much as possible, but the people in Italy and people in the United States, Canada, who's working in front line, they can see it, what's going on. So that's what we trying to see if it's true, test it ourselves, and then we're gonna get back to you on that. But yes, we had patients drinking cocoon water. We have a 89 years old patient living in an old folks home, and then he got, of course, COVID-19 infection, and he's back now from the hospital safely. There's no really a protocol. Yes, I can send you. There is a question here. There's no protocol on COVID-19. We have the protocol given by the association from Ayurvedic standpoint and natural medicine. This is an official release of that protocol. I can send that. And then cocoon water, yes, elevates CD4 and CD8 counts. That's the question. It is, yes, it is, elevates it every single time. And not just the CD cells, not just the CD3, also CD25 cells. And then there is a action of the natural killer cells. Is much better function after 14 days been drinking the water and taking the baths. The baths is very important too. The baths brings into the tissue large amount of oxygen. We are capable to elevate between 15 to maybe 20%, 25% higher than the patient arrives begin with. And if you think of this by, let's say, 14 days, pretty much we can help to get you in a better state of oxygenation in the tissue. So that's what it does. And that's what we see. So mainly is not the cure for the COVID-19, but I think there's three things. Liquid you need to have anyway. So you need to cool your temperatures. If you have fever, you have inflammation, you need to cool your temperatures. That's cocoon water is perfect. You need to stimulate your cellular immune system, perfect. If you need to reduce inflammation, perfect. And if you need to have solve the hypoxic issue, that's the best. So if all these four comes together at once, I think is very useful. And thank you very much, saying brilliant. Thank you for that. Thank you so much. So is that, Professor, can be proved by Drosophilus? It could be proved by this section, this 4 point I made?
Yeah, I mean, certainly we have the proofs regarding the expression of genes. I mean, the expression of genes significantly changed, significant expressional change, regarding some genes that are implicated in inflammation, in anti-inflammatory responses, in innate immunity. So we have this kind of and overcoming hypoxia. So mechanisms there will be promoted some mechanisms that would overcome the negative effects of hypoxia. That's quite interesting. I haven't mentioned to you, but I can tell you that these gene expression data are indicating that when it comes about the ATP productions, I mean, to produce more ATP because the cell is in need of ATP, this water would force the mitochondria, or would make even mitochondria to fuse, and you would have giant mitochondria formed to make the ATP production even more efficient. So there are so many amazing things that we have to now that we've got the through these gene expression things, we have data, we have some indications, some proofs that here they are, what is going to happen. Now we are going to look for these issues that we are predicting on gene expression data to happen, and once we have those things, we would have the direct proofs for all these things that I was telling you that you were saying earlier. So it's amazing. It's amazing. It's amazing.
Thank you so much. That's fantastic. Thank you so much. I hope we can amaze you more. I hope we can amaze you more, and we can bring more to the people on this. I would like to thank everybody coming tonight and be with us. And we will continue to do this. And next time I would like to talk about a little bit of wound healing. Maybe you can join up with me on that one. Come to the studio here, and I can show some of the wound healing effect of the oxygenation on hypoxia and normoxia issue, and that would be very interesting also. And maybe you could support that theory and that how the gene expression supporting this, what we're seeing at real life action.
Yeah, we'll see.
Okay.
I'm gonna try my best.
All right. Thank you so much. Good night, everybody, and good day for everybody.
Bye-bye.
Thank you. Bye-bye.
Bye.